All right, so today we're going to finish up our lecture talking about points of substrate or points of regulation for glycolysis versus gluconeogenesis. Yesterday we talked about how there are 3 sites of regulation to determine whether either glycolysis or gluconeogenesis are going to occur. And we talked about the first substrate cycle in regards to hexokinase and decided that, and talked about how this wasn't the predominant regulatory role. But let's revisit a little bit about what we talked about so that we can address each of the 3 substrate cycles in this talk. Okay, so substrate cycle 1 is defined by hexokinase or glucose 6-phosphatase when we're talking about gluconeogenesis. But again, we usually talk about this regulation in regards to the glycolytic enzymes. So glycolysis is on this side. So we're going to talk about that in terms of glycolysis. So the first substrate cycle is hexokinase. There's 4 different isozymes. If we build up glucose 6-phosphate, it causes a product inhibition, which causes inhibition of hexokinase. So too much of the glucose 6-phosphate is made, hexokinase is inhibited, and the rate of formation equals the rate of utilization, and we become a steady state. So at that point, a steady state, we have a futile cycle where no glycolysis or gluconeogenesis is occurring. We talked about why hexokinase was not the main regulatory enzyme. We gave 3 reasons. 1, that glucose 6-phosphate can come from glycogen breakdown. It's the major product when we convert glycogen to a single monosaccharide, so going from a polysaccharide to a monosaccharide. And so we can bypass the overall first reaction and bypass hexokinase to make pyruvate by breaking down and getting our starting material from glycogen. In addition, glucose 6-phosphate has 2 other pathways that it can be utilized in, the pentose phosphate pathway, or it can actually be used to make glycogen in the liver as shown right here. All right, so that's really the reason why hexokinase is not the main regulatory enzyme. All right, so substrate cycle 2, the phosphofructose kinase and fructose bisphosphatase is really the main regulatory reaction or main regulatory substrate cycle of glycolysis and gluconeogenesis. And this is where we stopped class on yesterday. Okay, so the regulatory flux is determined by the concentration of essentially allosteric effectors, and these can be either positive allosteric effectors in the case of ADP or AMP, or negative allosteric regulators in regards to ATP and citrate. We talked about how ATP is both a substrate and a negative allosteric effector, and we talked a little bit about why that was the case in regards to the 2 binding sites on the PFK1 enzyme. And we're going to show this again in just a minute. So what happens is, as represented by this graph, we have our concentration of substrate, fructose 6-phosphate. We have that we're measuring activity, and we get, as would expect, some sort of curve that looks like a Michaelis-Menten curve, where we have fructose, as we add substrate, we're getting increase of our phosphofructose kinase activity. Now this happens in low ATP, and so that means that our fructose 6-phosphate is in excess, we have low ATP, and they're both binding at this catalytic site. What happens is, once you start to saturate the enzyme with high ATP concentrations, exogenous or excess ATP is going to bind at this other site, this allosteric site. And once it starts to bind at this allosteric site, we're going to start to have a decrease in enzymatic activity. Okay, there's other regulators that we'll talk about in addition to citrate, AMP, ADP, and ATP, and that's in the allosteric effector fructose 2,6. phosphate. Sorry, bisphosphate. And we'll talk about that in just a minute. Okay, but first I want to show you the structure of this phosphofructose kinase 1 enzyme so that we can observe the catalytic site and the allosteric site. All right, so this is the structure of phosphofructose kinase 1 from E. coli. It's the crystal structure, and it's a 4 subunit protein, so it's a tetrameric protein where we have 1, 2, 3, 4. Now these subunits are all homologous, which means they all have a catalytic binding site, which binds our substrate, fructose 6-phosphate, bisphosphate, so fructose 1,6 bisphosphate, all right? And it also binds our ATP. You can't really see it, it's a little bit hard to see, but it also binds the ATP, all right? This catalytic site allows the ATP to be bound in close proximity to our fructose 1,6 bisphosphate. There's another site present, so we're just looking at the subunit 2, which is this kind of light blue, that's away from it, away from the catalytic active site, that binds those allosteric regulators, whether it's ATP, ADP, AMP, fructose 2,6 bisphosphate, all right? So this is a little bit complex for you to see because there's a lot going on, and so a better way to show this would be just through a cartoon, all right? And so we're looking again at the enzyme phosphofructose kinase 1. If you don't remember, this is the reaction that we're looking at, the conversion of fructose 6-phosphate to fructose 1,6 bisphosphate. All right, so we're adding the phosphate to the OH group on that methanol at the first position. All right, so let's talk about ATP binding. At low ATP, it will bind only to the active site because it is a substrate. This is a transferase that's coming from ATP, binds to active site. Remember, kinases are transferase enzymes. Not sure what's happening there, but they're transferase enzymes. So this phosphate group is coming from ATP, okay? And so at low ATP levels, it's binding to the active site shown by this Pac-Man or this cartoon shown here. So you have your fructose 1,6 bisphosphate binding as well as your one molecule or low amounts of ATP. This will stimulate glycolysis, which means that the fructose 1,6 bisphosphate, which means the phosphofructose kinase will be upregulated and glycolysis will occur. All right, what happens under high ATP conditions? Well, what happens is it'll bind to the active site. I'm not sure what's happening to the pen. Binds to both the active site and the allosteric site. All right, and so once the ATP binds to the allosteric site, we've then inhibited glycolysis and stimulated gluconeogenesis. This happens as a result of a conformational change on the enzyme. So conformational change causes inhibition. So that's really why we can have ATP be both a substrate and an allosteric regulator or inhibitor. Okay, so under high ATP concentrations, we inhibit glycolysis and we stimulate gluconeogenesis. Okay, so let's talk a little bit about, not sure why that's in there. Not sure really what happened there. All right, so let's talk a little bit about what happens with this other allosteric modulator, fructose 2,6 bisphosphate. So now we're adding at the second position, so shown here, fructose 2,6-bisphosphate, and this is catalyzed by the enzyme phosphofructose kinase 2. It's an isozyme of phosphofructose kinase 1, so they catalyze very similar reactions, but phosphofructose kinase 2 phosphorylates fructose 6-phosphate at this 2 position and not the 1 position. All right, so PFK1 adds P at 1 position. PFK2 adds P at the 2nd position. All right, phosphofructose kinase, or fructose 2,6-bisphosphate, is not a glycolytic intermediate. That means it's not made in glycolysis. It's made only by this PFK2 enzyme, which is not a glycolytic enzyme. All right, so what happens is fructose 2,6-phosphate will actually stimulate glycolysis, and that's kind of what this graph is showing here. All right, so stimulates glycolysis. All right, so under no fructose 2,6-bisphosphate, only fructose 6-phosphate, you have a normal activity shown here. So normal activity. Upon binding of the fructose 2,6-bisphosphate, you actually stimulate glycolysis. You increase the affinity of binding of the substrate. So increase fructose 6-phosphate affinity and activity. All right, that's what this graph is showing. So, fructose 2,6-phosphate stimulates PFK1, which stimulates glycolysis. It binds to the allosteric site. It inhibits the reversed reaction, which is fructose 1,6-bisphosphatase, so FBPase1, therefore inhibits gluconeogenesis. All right, there's one homework question that talks about cyclic AMP. So the cyclic AMP levels cause PFK2 inhibition. All right, so regulated and cause inhibition of PFK2 enzyme. All right, so if PFK2 is inhibited, that means no that means no fructose 2,6-bisphosphate is being produced, and that means that gluconeogenesis will actually be activated. All right, so that's actually asked on a homework question, how cyclic AMP affects, I think it's how it affects glycolysis or something like that. All right, and so that's also detailed in your textbook. All right, so Phosphofructose kinase is the main regulatory enzyme, or the main regulatory substrate cycle. That's important to know. It's important to know why ATP can both activate and inhibit this enzyme, and all the effects that those different allosteric effectors have in regards to increasing or decreasing glycolysis versus gluconeogenesis. All right, so the last substrate cycle, cycle 3, is pyruvate kinase. It is also allosterically regulated, but it's also regulated by covalent modification. All right, so let's talk a little bit about the allosteric regulations. And so ATP, acetyl-CoA, and long-chain fatty acids actually inhibit pyruvate kinase. Because that means that they are signs that there is abundant energy. And so if we have abundant energy, it means that we don't need to make more energy in the form of ATP, which again is one of the products of glycolysis. And so we inhibit pyruvate kinase, and glycolysis is inhibited. What is going on with this pen? All right, so fructose 1,6-bisphosphate actually activates pyruvate kinase. That means that more product is being built up from that third substrate cycle, and it's saying that yes, we need to activate glycolysis and therefore activate pyruvate kinase. Need more energy. Activate glycolysis. All right, the second way that pyruvate kinase, in addition to allosteric regulation, is by covalent modification. And this has to do with the phosphorylation of a active site residue, I believe it's a histidine, that will inactivate pyruvate kinase. All right, so the phosphorylation inactivates pyruvate kinase and therefore slows down glucose use. Or glycolysis. Okay, and so that's essentially what this diagram is showing. If we have pyruvate kinase that's phosphorylated, it's inactivated, and we'll essentially make no ATP. All right, L means lung. There's a lung and muscle form. Once it's dephosphorylated, the enzyme will become activated, and it can undergo these different covalent modifications. Regulatory roles shown here. We've got our allosteric effectors shown here. And this can also be, like I said, regulated by the product of one of our other steps, 6 steps ahead in the third substrate cycle. So again, that's another reason why that substrate cycle 2 is such a main point in regulatory, because it also has downstream regulatory effects here at the substrate cycle 3 in the levels of concentration of fructose 1,6-bisphosphate. All right, just a few more slides. I want to put this a little bit into context for hormone regulation. All right, so this is actually in chapter 23, but there are some questions on the final exam, as well as the sapling, in regards to how glycolysis and gluconeogenesis and regulation are affected by hormone control, mainly in regards to the hormones insulin and glucagon. All right. So let's talk a little bit first about insulin. So insulin is a hormone. It's a small peptide. It's one of the very first sequences that were solved undergoing the peptide sequencing that we talked about when we talked about amino acids. And so insulin is a small peptide hormone made up of different amino acids, and it's released by the pancreas. And it's actually released by pancreatic beta cells. That's a homework question, beta cells. So it asks if it's secreted by alpha or beta pancreatic cells, and it's actually released by pancreatic beta cells. All right, so insulin is released under high glucose. It favors the conservation of blood glucose in two ways. All right, and so these blue arrows are showing essentially what happens when we have high glucose. And high glucose means greater than 5 millimolar. All right, so we've got large amounts of glucose in the blood. What happens? The blue arrows represent glucose. Okay, and they're showing the different fates of glucose. All right, so we want to start to conserve blood glucose, which means to increase glucose uptake. All right, so essentially we're favoring the conservation of blood glucose two ways. One is to make more glycogen synthesis in the liver or the muscle, so increase the amount of glycogen that's being produced. All right, so in the liver and the muscle, increase glycogen synthesis, and that's shown right here. So number one. The second is to start converting your glucose into fat storage. And again, we didn't really talk about that, but that's shown here in number two. Increase the synthesis of triacyl glycerols, and we've talked about what triacyl glycerols are. They're energy storage, and we talked about how they are more sufficient at storing energy than glycogen. All right, so these two things result in an overall lowering of the blood sugar. Back to normal and homeostasis. All right, so we want to make more glycogen, make more fat, and it causes, if we make more glycogen, we're going to decrease glycogen breakdown. And again, increase glucose uptake. And there is a question on this on the sapling homework as well. All right, so this is the result of insulin release. So this is under a high blood sugar state. What about under, release, not result. What about under low blood glucose? So under low blood glucose, another peptide hormone, glucagon, is released. It's also a peptide made up of amino acids similar to insulin, but it is released under low blood glucose. And low blood glucose is anything less than or around 4 millimolar. So this is not really a whole big change in concentration between what the body considers a high amount of glucose and a low amount of glucose. All right, so at this point we want to make more glucose, so we want to increase blood glucose concentration in several ways. All right, we want to break down glycogen. This is known also as, it's on the glycogen slide, glucogenolysis. There's a homework question on this. So it's going to favor the breakdown of glycogen in the liver. And so here we've got our glucagon release down to the liver. We're going to break down glycogen, all right, to make more glucose 6-phosphate, to essentially make more glucose to bring up our blood sugar. We're going to stimulate gluconeogenesis. So we're going to break down more glucose. We're going to make more glucose from gluconeogenesis. And we're going to therefore have a result in overall higher blood sugar, or increased blood sugar levels. The primary target for glucagon release is the liver. However, there is release in glucagon both in terms of the fat tissues, adipose tissues, that's shown here, and in hormone receptors in the brain. All right, and the main hormone receptor is, or one of the predominant hormone receptors is epinephrine, and both of these signals and hormone regulations, the fat release or epinephrine signals, will stimulate the liver to begin breaking down glycogen and releasing more glucose 6-phosphate to make more pyruvate and therefore make more energy. All right. All right, so with that, that's the end of the regulation chapter. So we talked about some common features of regulation in regards to metabolic pathways. We talked about some features of regulation. Make sure you understand how to relate Q and K, both numerically, remember 1 to 2 orders of magnitude, but also if we ask you to solve for Q and K to see if a reaction is at equilibrium or not, you should be able to tell whether it is. Make sure you understand the 3 substrate cycles that we talked about, the major point of regulation being that substrate cycle number 2 in phosphofructose kinase. Understand how both the concentrations of substrates and products, allosteric and covalent modifications can regulate whether glycolysis or gluconeogenesis is going to occur. Do we need more pyruvate or do we need more glucose for energy? And then finally, understand a little bit about hormone regulation in regards to insulin and glycogen, where these are occurring, this hormone regulation is occurring, and how it relates to either increasing or decreasing overall blood sugar and regulating that effect. Okay, thank you very much. Remember, we'll have an in-class assignment covering not only regulation, but all of the material on metabolism on the Monday after Thanksgiving. Happy holidays, and I'll see you all then. Bye.